Welcome, students, to the first of several chapters about the nervous system, which is exercise 15, kind of just an overview of the nervous system. In later chapters, we'll get into special senses and the brain and the spinal cord and cranial nerves and just all the other parts of all of the parts of the nervous system. But this chapter really is just going to start with an overview. So we break the nervous system into two main parts, what we call the central nervous system and what we call the peripheral nervous system. Now, the central nervous system consists of the brain and the spinal cord. The peripheral nervous system consists of pretty much everything else. So all of the nerves, all of the nerve clusters, the ganglia, all of the sensory receptors, all of that falls into the peripheral nervous system. Now, the job of the nervous system is to take information kind of from the body and from the external environment, kind of integrate it and put some sort of output. It's the controlling system of our body. And there are two broad categories of neural cells, what we call neuroglia and what we call neurons. So the neurons are the functional cells. They're the ones that are actually transmitting all of the signals, allowing the nervous system to kind of be the boss of your body. The neuroglia are support cells. They're helper cells. And they are different depending on whether we're in the central nervous system or in the peripheral nervous system. There are more neuroglial types in the central nervous system than there are in the peripheral nervous system. In the CNS, we have astrocytes, oligodendrocytes, microglia, and ependymal cells. So the astrocytes are kind of the most abundant of the CNS neuroglia. They play a lot of different roles. They form part of the blood-brain barrier to protect your brain tissue. They recycle neurotransmitters. They monitor kind of the ion environment around ions. They guide the growth of young neurons. So they really have a wide variety in terms of their functions. Oligodendrocytes, their job is to produce myelin, which is insulation. Microglia are immune cells. The rest of the body's immune cells have a very difficult time accessing the brain and spinal cord tissue, and so the central nervous system has their own set of immune cells that help kind of defend us from things that might potentially do us harm. And then the ependymal cells, their job is to secrete what we call cerebrospinal fluid, and then also help circulate it through the central nervous system. In the peripheral nervous system, we have Schwann cells, which are the ones that make the myelin, and then satellite cells, which function very similarly to the astrocytes in that they have a lot of jobs. So you can see here is an astrocyte kind of wrapped around some capillaries in the brain, preventing stuff from getting out of the blood and into the brain. See a microglia here, kind of monitoring these neurons, making sure that they're healthy and not infected. Ependymal cells here that are going to make this CSF, and then because they have cilia, can help circulate it through the central nervous system. The oligodendrocyte here, which creates this kind of insulating fatty substance called myelin. And then in the peripheral nervous system, we have the Schwann cells for creating myelin. And then the satellite cell, which is kind of this overall support cell that surrounds the cell body of our neuron. So if we look at our neurons, we tend to break neurons into kind of two parts: the cell body, and then these processes that hang off the cell body. The cell body is the gray matter, if you've ever heard that term. And the way the nervous system organizes is that cell bodies of neurons tend to be together, and axons of neurons tend to cluster together. In the central nervous system, clusters of cell bodies are known as nuclei. Now that's different from like the nucleus of a cell. We're talking about clusters of the cell bodies of the neurons. In the peripheral nervous system, those clusters have a different name. They are called ganglia, or ganglion, singular. If we look at the cell body, you'll find the nucleus. That's like kind of the control center for the whole neuron. Neurofibrils, which help give the neuron some shape. And then Nissl bodies, or chromatophilic substances, which are part of that metabolic machinery. Because the job of the cell body is to be the biosynthesis center. It is responsible for everything that has to get made in terms of different proteins that we might need, ion channels that we might need, any lipids that we might need, anything that has to get made by a neuron is going to get made in the cell body. And then the processes fall into one of two types. The dendrites, which receive signals from elsewhere, and then the axons, which generate and conduct signals through the nervous system. Now, clusters of axons are known as white matter, and they're called tracts in the central nervous system and nerves in the peripheral nervous system. The way the signals kind of move through the axon is they get started at the axon hillock, that's where they're generated. They travel down the length of the axon to the axon terminal, and then the axon terminal secretes neurotransmitters to keep the signal going to kind of the next cell in the chain. So if we look at a neuron, you can see here is your cell body, or soma, S-O-M-A, which is kind of the biosynthetic center for our cell, the dendrites for receiving signals from cells, and then the axon. The axon hillock here is where the neural impulses are generated. Those impulses then travel down the body of the axon to the axon terminals, which are our secretory regions of the axon that are going to release these neurotransmitters that allow us to kind of take that signal from one neuron and send it to another neuron or to some other effector cell. Now let's talk about myelination. Now, myelin is basically this kind of fatty lipoprotein substance that acts like an insulator. So it wraps around these axons kind of like a jelly roll or a Swiss cake roll, if you're a big fan of Swiss cake rolls like me. But not the whole of the axon is wrapped in the jelly rolls. There are portions of the axon that don't have myelin. They're called nodes of Ranvier or myelin sheath gaps. Now, the two types of neuroglial cells that make myelin are Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system. And they do it in a slightly different way. In the peripheral nervous system, each jelly roll that you see is essentially one Schwann cell that is wrapped around the body of the axon. Whereas in the central nervous system, one oligodendrocyte can make several jelly rolls around one axon or around many axons, and so they have a slightly different kind of secretory or production process, and same end result with these jelly rolls of kind of fatty myelin to act as an insulator to help our signals travel faster. We also then have synapses, which are the connections between either neural cells. So if we were in the central nervous system, we're going to connect one neuron to another neuron. Or if we're in the peripheral nervous system, we might connect a neuron to a muscle or a gland or something like that. You've seen synapses before when we looked at neuromuscular junctions. There is no physical contact. There is a little fluid-filled space called the synaptic cleft, separating kind of the presynaptic cell, the one is sending the signals towards the synapse, and then the postsynaptic cell, which is kind of the one receiving the signals from the synapse. We can classify neurons based on their structure or shape. They have slightly different shapes. They fall into one of three types: what we call multipolar neurons, bipolar neurons, and unipolar or pseudo-unipolar neurons. The multipolar neurons tend to have very discrete dendrites and axons, where they have these really long axons that are quite distinct from the dendrites. You can see axon, lots and lots of dendrites. Axon, lots and lots of dendrites. Bipolar cell with the cell body kind of at one end. The cell bodies of bipolar neurons tend to be kind of smack in the middle with the two sets of processes hanging off either side, and it's a little more difficult to determine which is the axon and which is the dendrite. And then in unipolar neurons, really what you have are the cell bodies off to the side with the processes excuse me, hanging off, and it is very difficult with unipolar neurons to kind of tell if you were just looking at it kind of on its own, which is the kind of dendrite, and which is the kind of receptive area, because it's just kind of this all one long central process. Now, we can also classify neurons by their function. Are they taking information into the central nervous system, or are they bringing information out from the central nervous system? So afferent or sensory neurons are bringing information from the other parts of the body into the central nervous system. They are typically unipolar. So you can see here's a little cell body with the central processes hanging off. Efferent or motor neurons are usually multipolar. They are taking kind of motor data, how to move a muscle, what this gland should do, from the central nervous system out to the body. And then interneurons are association neurons that are found in the central nervous system that will allow neurons to connect to one another. We can also then look at, in exercise 15, some nerve structure. So nerves are found in the peripheral nervous system. They're bundles of axons, basically. They're these very cord-like organs. They have connective tissue coverings, very similar to what we saw with the muscles. And nerves are taking information from the central nervous system out to every other organ in the body, out to the different digestive viscera and the muscles and the endocrine glands and the skin, and et cetera, et cetera, et cetera. Now, nerves can either be sensory, motor, or mixed. Sensory nerves are only carrying kind of afferent information into the central nervous system. Motor nerves are only carrying kind of efferent information out of the CNS to a particular organ, maybe a muscle or gland. But most of the nerves of the body are actually mixed nerves. So bundles of axons, some of those axons are going to be carrying sensory information, some of those axons in that bundle are going to be carrying motor information, which is kind of a much more efficient use of the space there. So if we look at a cross-section of a nerve, you'll see that there are 3 connective tissue sheaths, very similar to what we saw when we looked at muscles. You can see here is a single axon surrounded by its myelin sheath. That whole axon with its myelin sheath then gets covered in endoneurium. We get clusters of axons together, and we have a fascicle that gets covered in perineurium. We get several fascicles bundled together into a whole nerve, which gets then covered in epineurium. So again, very similar to what we saw with skeletal muscles. And that is the end of this chapter.